Cell and gene therapies represent groundbreaking advancements in medicine, offering targeted treatments by directly modifying or replacing genes within a patient's cells. These innovative approaches provide new hope for patients with previously untreatable conditions, enabling personalized treatments that can target the root cause of a disease at the genetic or cellular level. Key aspects of GMP in cell and gene therapy manufacturing include cell collection, genetic modification, and re-infusion. Each step requires stringent protocols to ensure consistency, safety, and effectiveness. Proper training and controlled environments are essential to maintain the quality of the final product. Sterility assurance in manufacturing is critical. This involves maintaining sterile environments, designing facilities to meet specific sterility requirements, and ensuring personnel hygiene. These measures help prevent contamination and ensure the safety of cell and gene therapy products. Environmental monitoring practices are essential to ensure cleanroom sterility. This includes air and surface sampling, equipment testing, environmental sensors, and personnel monitoring. These practices help maintain the highest standards of cleanliness and functionality in the manufacturing environment. Sterility testing for final products involves microbial testing, endotoxin testing, bioburden testing, and various test methods. These tests ensure that the final product is free from harmful pathogens and safe for patient use. Comprehensive sterility controls include safety and purity assurance, quality assurance, and thorough documentation. These practices ensure that cell and gene therapies meet the highest standards of safety, quality, and regulatory compliance. Traceability in manufacturing involves documenting every step, ensuring accountability, and managing recalls and investigations. This ensures that any issues can be quickly identified and addressed, maintaining the integrity of the manufacturing process. Validation and verification processes ensure that high-quality, safe products are consistently produced. This includes process validation, equipment and material verification, and personnel qualification. These steps are crucial for compliance with GMP standards. Quality control checks are essential at every stage of production. This includes raw material testing, intermediate product testing, and final product testing. These checks ensure that all products meet the required standards before reaching patients. The importance of GMP for safety and approval cannot be overstated. Consistency, preventing variability, and ensuring patient safety are key aspects. GMP practices ensure that cell and gene therapy products meet the highest standards of quality and efficacy. Facilitating regulatory approval involves meeting regulatory standards, comprehensive documentation, and inspection and audits. These steps ensure that products meet the necessary requirements for approval and can be safely used by patients. Enhancing patient safety involves minimizing contamination risks, ensuring sterile, safe, and effective products, and improving therapeutic outcomes. GMP practices ensure that therapies are both safe and effective for patients. Building trust with stakeholders is crucial for the success of cell and gene therapies. This involves ensuring patient confidence, healthcare professional trust, and regulatory and industry partnerships. GMP compliance helps build this trust. Airflow in cleanroom design is essential for maintaining sterility. This includes controlled airflow systems, HEPA filtration, positive pressure, and air changes per hour. These measures help protect the cleanroom environment from contamination. Materials and equipment in cleanrooms must meet strict sterility requirements. This includes using sterile materials, durable equipment, and preventing contamination. These practices ensure the integrity of the manufacturing process. Segregation in cleanroom design involves dedicated areas, controlled access, and physical barriers. These measures prevent cross-contamination and ensure product integrity throughout the manufacturing process. Environmental control in cleanrooms is crucial for cell and gene therapy manufacturing. This includes temperature control, humidity management, and particulate monitoring. These controls ensure optimal conditions for production. Personnel movement in cleanrooms must be carefully controlled to prevent contamination. This includes controlled access, hygiene protocols, logical flow design, and training and certification. These measures ensure a sterile environment. Material flow in manufacturing involves raw material entry, controlled pathways, labeling and tracking, and material storage. These steps ensure that materials are handled properly to maintain sterility and quality. Waste management in manufacturing includes waste segregation, secure disposal protocols, equipment decontamination, and waste tracking. These practices ensure compliance with regulations and prevent contamination. Commissioning is the crucial first step in the CQV process, ensuring all systems and equipment function as intended. It begins with system installation verification, confirming proper setup and connections of all components. This is followed by functional testing, where key systems like HVAC and air filtration are checked against designed operational parameters. The final stage involves equipment performance testing, where production equipment such as incubators and bioreactors are tested under real conditions. This comprehensive approach ensures that the infrastructure fully supports the desired manufacturing conditions, laying the foundation for safe and effective cell and gene therapy production. Qualification is a critical step in the CQV process for cell and gene therapy production. It ensures that cleanroom environments meet defined specifications, focusing on environmental and system requirements. This process is essential for maintaining compliance and is essential for maintaining product safety and sterility. Key activities include monitoring environmental parameters, calibrating equipment, verifying cleanliness standards, and conducting airflow and pressure tests. These steps are crucial for compliance with regulatory standards and preventing contamination in critical production zones. Validation is a comprehensive process that ensures manufacturing processes are stable and reproducible. It verifies that cell and gene therapy products meet specifications for potency, purity, and sterility under actual production conditions. This involves repeated production cycles to confirm consistent quality standards. Performance qualification tests the entire manufacturing system under operating conditions, ensuring consistent production of desired quality products. Long-term monitoring ensures ongoing performance and compliance, while regulatory compliance involves compiling and reviewing validation reports for regulatory approval. Design qualification, DQ, is the crucial first step in cleanroom qualification. It involves reviewing the design and layout to ensure compliance with required standards and specifications. This stage is vital for identifying potential issues, verifying suitability for intended manufacturing processes, and identifying necessary modifications. The process includes design review, compliance check, system specifications, and risk assessment to ensure the cleanroom design aligns with regulatory guidelines and GMP requirements. Installation qualification, IQ, is the first phase where physical equipment and systems are tested for compliance with design specifications. It ensures that all systems and equipment required for the cleanroom's operation are properly installed and function as specified. This involves verifying that all equipment is installed correctly, reviewing documentation, confirming physical presence and connections, and ensuring proper calibration and setup. IQ is a verified installation of all systems and equipment, supported by comprehensive documentation confirming the installation of all components. Operational qualification, OQ, involves testing the cleanroom and its systems under actual operational conditions. The goal is to confirm that the cleanroom environment can be consistently controlled and maintained according to predefined operational criteria. This crucial step ensures all systems perform as expected in real-world conditions. It includes environmental monitoring, system performance tests, functionality testing, and data logging and analysis to ensure operational parameters remain stable and meet required specifications. Performance qualification, PQ, is the final stage in ensuring cleanroom readiness for cell and gene therapy production. It involves running the facility under typical conditions to verify consistent performance and the ability to meet production standards. This crucial phase confirms the cleanroom's ability to reliably produce high-quality products while adhering to sterility requirements. During PQ, teams simulate real production runs, closely monitor quality control parameters, evaluate long-term performance, and implement continued monitoring practices to ensure ongoing compliance. FDA plays a crucial role in regulating cell and gene therapy products in the U.S. They provide comprehensive guidance documents for developers, outlining standards for product development, clinical trials, manufacturing, and approval processes. Preclinical testing requires demonstrating safety and effectiveness in animal models before clinical trials can begin. Clinical trials oversight ensures safety and efficacy through investigational new drug applications. Regulatory approvals involve safety, efficacy, and manufacturing processes before granting commercialization approval. Post-market monitoring ensures ongoing safety and effectiveness of therapies after approval. The European Medicines Agency, EMA, offers a centralized approval process for cell and gene therapies, allowing manufacturers to market their products across the entire EU once approved. This streamlined approach reduces barriers to innovative treatments. The EMA enforces strict guidelines for clinical trials under the Clinical Trials Directive, ensuring patient safety and product effectiveness. Additionally, cell and gene therapy manufacturers must adhere to good manufacturing practice standards, guaranteeing controlled, consistent, and safe production. Special incentives are offered for gene therapies treating rare diseases, including expedited review and market exclusivity. The World Health Organization, WHO, develops and promotes international standards for the quality, safety, and efficacy of cell and gene therapies. This includes producing reference materials and methodologies for testing, ensuring consistency across borders. WHO collaborates with countries and health organizations to establish and harmonize GMP guidelines for cell and gene therapy production. This effort aims to create a unified approach to maintaining quality control. WHO also provides ethical guidelines for the development and use of advanced therapies, ensuring that cell and gene therapies are developed and used responsibly. Anvisa, Agência Nacional de Vigilância Sanitária, is Brazil's health authority regulating cell and gene therapies. It enforces national regulations, including guidelines for clinical trials, pre-market approval, and ongoing monitoring of gene and cell therapy products. Anvisa ensures strict GMP standards in the production of cell and gene therapies, protecting public health and fostering innovation. Clinical trials involving gene and cell therapies require Anvisa approval, adhering to strict guidelines for safety and efficacy. Anvisa also oversees product registration, evaluating therapies for safety, efficacy, and quality before they can be sold in the Brazilian market. TGA demands evidence of safety and efficacy from preclinical and clinical trials before approving therapies for human use. Strict GMP standards are enforced in manufacturing, ensuring high-quality control and safety standards for all products. Cell and gene therapies must be registered with TGA before marketing in Australia, with assessments on quality, safety, and efficacy. Post-market surveillance continues through adverse event reporting and other programs after product approval. SOPRA enforces regulations requiring cell and gene therapies to undergo rigorous clinical trials, pre-market evaluations, and GMP adherence before commercial approval. This ensures high standards of safety, efficacy, and quality control. SAHPRA mandates approval for clinical trials involving gene and cell therapies. This process includes assessing trial design, ethical considerations, and patient safety to ensure responsible research practices. After approval, SAHPRA continues to monitor the safety and effectiveness of therapies in the market. The agency may take regulatory action if safety concerns arise, ensuring ongoing protection of public health. Cleanroom designs that fail to meet specifications for controlling environmental factors like airflow, temperature, humidity, or particulate contamination can lead to FDA warning letters. These deficiencies jeopardize the sterile environment crucial for cell and gene therapy production. Warning letters may result from failing to implement proper contamination prevention procedures. This includes not maintaining required cleanliness levels or using inappropriate materials and equipment for sterile conditions in cell and gene therapy production. Lack of continuous monitoring or inconsistent testing of cleanroom conditions, such as airflow or particle counts, can lead to warning letters. Proper environmental monitoring is critical for maintaining the sterile conditions required for cell and gene therapy production. Failure to document critical steps in the manufacturing process, including raw material sourcing, process parameters, testing, or quality control checks, can result in regulatory warnings. Inconsistencies or deviations from validated processes, such as changes in manufacturing methods or failure to follow established procedures, may trigger warning letters from regulatory bodies. Adhering to standard operating procedures, SOPs, is crucial for maintaining product quality and safety. Lack of traceability or poor record keeping can lead to warnings. Traceability is vital for ensuring that the manufacturing process can be reviewed if a safety issue arises. Proper record keeping allows for efficient problem-solving and quality assurance. Cleanrooms may fail to meet stringent qualification standards due to incomplete design qualification, DQ, installation qualification, IQ, or operational qualification, OQ, processes. This can lead to inadequate performance and regulatory non-compliance. Staff operating cleanrooms or manufacturing equipment without proper training or certifications can violate GMP standards. This is a common reason for regulatory action. Failure to conduct regular qualification testing or re-qualify after modifications can prompt regulatory warnings. Cleanroom qualification should be an ongoing process to ensure the environment remains within required standards. Non-compliance with regulatory requirements may significantly delay product approval, impacting the company's pipeline and future projects. Repeated violations could lead to severe consequences, including product recalls or, in extreme cases, suspension of manufacturing licenses, effectively shutting down operations. Regulatory agencies may conduct more frequent inspections and audits, leading to higher operational costs and additional scrutiny on future products. The public and healthcare community may lose faith in the ability to produce safe and effective therapies, impacting long-term business prospects and partnerships. For cell and gene therapy manufacturers, adhering to GMP and regulatory requirements is critical for patient safety and maintaining trust across the healthcare ecosystem. Proactive compliance helps avoid serious consequences associated with regulatory warning letters.